Isaac Alonso from Mr. Lima's AP Biology class, and this is my PowerPoint on lipase. First of all, lipase is an enzyme, but in order to talk about it, we must first understand what an enzyme is. An enzyme is also referred to as a catalyst, which speeds up a reaction in order for it to occur more rapidly. Lipase is an enzyme, however, like any enzyme, it has its own function. And this particular enzyme is responsible for breaking down the fats that are found in the food that we consume in order for our inner intestines to absorb it. The structure of lipase, which can be seen in the image to your right, even though this image is a pancreatic lipase, however, not all lipase have the same structure. But what can be found in all lipase is an alpha hydrolysis fold. Also, lipase, even though they may not have the same structure, they still share to another common thing. You can say that lipase is separated into two sections: the N-terminus, which contains the alpha hydrolysis fold and the active site, and the C-terminus, which contains two beta sheets. Both these sections, which are connected together as seen in the picture, is connected by a single peptide bond. Also, the alpha hydrolysis fold, which is found in common in all lipase, is the keystone for its function. The reason all lipase have this is because it is responsible for the catalytic function it exhibits. The image seen before, as you can see, is split into two parts, and in the middle is where the active site is found. There are also different kinds of lipase. For instance, the picture shown before was a pancreatic lipase, which is found in the pancreas and is secreted. However, it cannot work without the help of colipase. Then there is lipoprotein lipase, which is responsible for cells in our blood to take the fatty acids in order to break them down. It can be said that lipase has gone through some sort of evolution because of the pancreatic lipase as shown before. Like earlier stated, it needed the help of colipase in order to function. However, pancreatic lipase must have gone through some sort of trial and error before realizing it needs the help of colipase in order to function. In addition, after several gene duplications, these enzymes took different roles based on their expression patterns and sequences. Lipase can be used for various reasons. In today's age, many companies have been using lipase in everyday needs such as baking, laundry detergent, and as its true role as a biocatalyst. These enzymes are also used to replace other enzymes, and they can be used for different functions, and in many cases they're even safer than others. The DNA of lipase, even though the structure of lipase may be different, they all still share the same DNA, which has the same function of breaking down fatty acids. In other words, you can say that this DNA is conserved in all lipase. DNA can also be found in the most common place for all lipase, which is the alpha hydrolysis fold. Then there are organisms who live in extreme conditions. These certain organisms are known as extremophiles. One of the most common extremophiles are microbes, which are able to adapt to its environment and thus survive in it. Lipase is also an example of an extremophile. The reason is because lipase was derived from a particular group of extremophiles, so it is able to adapt just as well to live in any sort of conditions. This concludes my PowerPoint on lipase, and leave any comments you have below.